Pina, and I'm the Executive Director of the CIBM Center for Biomedical Imaging. And we've been organizing these breakfast and science seminars. Actually, Mary Bach is the one who initiated them more than 10 years ago. And it's my great pleasure to carry on the torch for Mary and the community. And this is really a place where we share our latest research and questions that we may have for the community to join the groups, or to really things that you don't know, and to reach out. So it's really an opportunity to share. And last year we focused this breakfast and science seminars on CIBM core members. And this year we wanted to focus more on the affiliate members, meaning the partners of the founding institution partners, researchers. And so this year, during the summer, we said, why not give a chance to the promising researchers of the future and leaders of the future, which are PhD students and very precious. And so we thought that the summer edition of the Breakfast and Science Seminar would be giving an opportunity to the PhD students to share with the community. And so today, it is my great pleasure to introduce two PhD students. And the second speaker today will be Hamza Kebiri, who is in the Medical Image Analysis Lab at the University of Lausanne. And he is supervised by Dr. Marichel Bach-Quadra, who is one of the CIBM section heads for signal processing at UNIL. And so he will be sharing with us towards super resolution reconstruction of the fetal brain MRI. So, okay, thank you, Hamza.
Thank you, Pina, for the introduction. So, good morning, everyone. So, the second talk will be about this super resolution reconstruction of the fetal brain from diffusion MRI. But before going into the topic, so I will just give a brief introduction about the context, and then I will talk about this quantitative FETAL study that we have performed, and also the super resolution enhancement with an autoencoder. Okay, so as you know, so the brain can be decomposed into white and gray matter. So white matter is basically all these myelinated axons that link the different cortical areas in which the action potential can flow. And basically this white matter can only be depicted today with in vivo and non-invasively with diffusion MRI. So basically this technique relies on the diffusion of water molecules, and it uses it as a proxy to basically reconstruct the underlying structure of the brain. And it is particularly important, especially for the fetal brain, where actually it can help to assess the brain maturation, both for healthy and pathological subjects. And also, it can also actually help to reconstruct, to perform basically connectomics, basically this discipline that aims to link the different brain regions. And also from a clinical perspective, it can also be used to detect ischemia and edema. So for the fetal brain, unfortunately, we have a very low SNR and low resolution, and this is actually due to the fact that the fetal brain is very small, and it is embedded in the mother womb, basically surrounded by all the maternal structures and the amniotic fluid, and also to its distance to the coils. So in the end, by having this low SNR, we can only have a low resolution. And also there are significant artifacts linked to the diffusion sequence, mainly the distortions, which are pretty high using this echoplanar imaging sequence, basically this fast switch on and off of the gradients. And also one of the biggest problems with fetal MRI is also this fetal movement, which is usually random, so it cannot be modeled before. And also to minimize the mother discomfort, so we can only use small b values and the small number of gradient directions. So here comes the super resolution, where actually it can help to increase the resolution of our images, either so by basically smartly merging different orthogonal views, or only by using a single view and then enhance it, for instance, using machine learning. Okay, so our images look like this. So on the left, we have the B0, and on the right, we have B700. This data comes from the Zurich Children's Hospital. It was kindly provided to us by Andras Jakob. And as you can see, it is pretty degraded. Okay, so basically we work with these volumes, and for instance, we have this. We have basically our 4D volume here, which is composed of different 3D volumes. Each 3D volume corresponds to one gradient direction, and in the end, for our specific case, we have 15 directions, so we have 15 3D volumes. And we actually acquire many 4D volumes in different and orthogonal orientations. So for instance, it can be 3 axials and 2 coronal acquisitions. And so afterwards, we pre-process this data by first of all denoising it and then correcting for the bias field. So these slow intensity changes in the scanner, and then we correct for the distortions and also for the motion using the structural image, which is super resoluted. And afterwards, we merge the different orthogonal series. Okay, so now I will talk about this quantitative FETAL study that was accepted in the computational diffusion MRI workshop, and basically its goal is to since we have this limited scanning time, we actually want to quantify this trade-off between the gradient direction. So basically the number of the dots that are sampled here from the sphere, which is the number of these 3D volumes, and also the number of orthogonal series. Since in the clinical routine, one has to make this kind of choices. Okay, so we basically use this crossing phantom here, which is which has these polyester fibers, which are of 15 micron diameter, and we have this customized FA fractional anisotropy of 0.6 in the single fiber region. And in the crossing area, we have an angle of 60 degrees, which is basically here in this image here. Okay, so we actually acquire 2 sets of acquisitions. So the one on the left basically corresponds to the FETAL acquisitions, from which we want to derive all the diffusion metrics, and then we want to compare them to high resolution acquisition and see how good are we doing. So we typically use a B700 for the FETAL acquisition, and also we vary the gradient directions from 9, 16, or 25, and we acquire several orthogonal stacks. And for the high resolution volume, we actually acquire it in a 3 Tesla scanner, and we consider this B700 and 61 directions as our pseudo-ground truth to which we will compare our work. Okay, so the images look like this. So we basically have our crossing here, and we will mainly see this coronal slice in the next slides. Okay, so this is basically the framework of the work. So we on the right we have the FETAL acquisitions, and on the left we have our pseudo-ground truth. So we pre-process them both, and then we use 3 configurations to compare for the low resolution series. So either we just reconstruct the different metrics using the one volume, so it can be only one axial or one coronal, one sagittal, or by using a paired configuration, meaning we co-register both, for example, coronal sagittal or coronal axial, sagittal axial. And the third one is where we merge the 3 series basically, and we use the super resolution reconstruction here. But we actually use it only for each 3D volume, so we don't account here for the angular dimension. Okay, so basically our regions of interest are here. So we delineate the single fiber area here in orange, and in blue we have the crossing area. And from the tensor, we reconstruct the fractional anisotropy and mean diffusivity. So these are basically measures of diffusion. For instance, fractional anisotropy quantifies how much the diffusion is along one dominant direction, So for instance, it will be very low in the CSF and the cerebrospinal fluid, and it will be high, for example, in the white matter, like in the corpus callosum. Also compute the mean diffusivity, and also we reconstruct the fiber ODF using the constraint spherical deconvolution algorithm, from which we derive the fiber orientations. So these are the first results. So the first, what we noticed first is that the fractional anisotropy that was provided to us by the vendor is different from the one that was empirically found, and especially here for these other volumes. And also we can actually see here the ground truth in the orange, in the horizontal lines, where the orange means single fiber area and the crossing is in blue. And basically, the higher the disk radius, the higher the number of direction is used. So we can see that we have a slight increase in accuracy with the number of directions, but not that much. And also we can see that the more we combine the stacks, the more we somehow tend to be closer to the ground truth. And for the mean diffusivity, so we could reconstruct the mean diffusivity that was provided to us by the vendor, around 1.2. And we can see that the number of gradient directions do not influence the accuracy of the reconstruction. And also merging different series doesn't seem to help as well. So we also reconstructed the fiber orientations, but for the sake of time, I won't present them here. But we also find a similar results. So where basically the number of gradient directions do not influence a lot the accuracy of the measurements. But you may ask why we didn't actually reconstruct the fiber crossings. This is mainly because we couldn't actually reconstruct them a lot, so we tried, but the main problem was probably the low B value, which was at B700. So unfortunately, we could only see very few of them as shown here. Okay, so to conclude this first part, so we have seen that fractional isotropy is more sensitive to diffusion directions than the mean diffusivity. But maybe the take-home message here for this part is that more orthogonal series are give basically a better accuracy over the number of gradient directions, especially in this experiment here. And also we can hardly actually reconstruct the different crossings at B700. So the main limitation of this study is that we don't use a motion here, which is the main caveat in fetal imaging. So these results can be actually taken as an upper bound that can be reached. So the next step is to apply this motion, basically to include motion in our study, and also to apply this similar pipeline to other phantoms and different scanners. And then we plan to submit this work to the Medical Image Analysis Journal. Okay, so now going to the second part of the talk. So basically, the goal of this part is basically to enhance the different acquisition, like by using this autoencoder. And if we get the big picture here, is basically to so after the pre-processing, we want to take each single for the acquisition, and then we want to increase the true plane resolution, so the zero resolution. And we have actually the hypothesis that this will result in a better reconstruction in the end, also through the fact that motion will be estimated better. So basically, we take one single slice, and then we try to do the super resolution here by basically enhancing this the true plane resolution. And so one can actually think about just interpolating. So for instance, if we have here a 1 by 1 by 4 cubic millimeter, and we want to go to 1 by 1 by 2 cubic millimeter, we can just interpolate. But since today we have machine learning, we can see the problem as an image synthesis problem. And one can basically use all the amounts of data to extract some kind of patterns and see hopefully that it will generalize to new unseen data. So especially for our case here, we use an autoencoder, which is an unsupervised technique or self-supervised technique, which means that we don't provide any label here. So it has the machine learning methods have this advantage of not putting any hypothesis on the images. And for our case, we used an autoencoder, but we could also have used like a GAN or simply a cascade of convolution neural networks. We basically choose these autoencoders because they are simple and intuitive, and also they were proven to work quite well in the cardiac MRI by basically synthesizing this average slice. And basically cardiac MRI also have the same problem of thick slices. For instance, in this example, so the slice thickness was about 7 millimeters. Well, for our case, we have like a 4 millimeter, 5 millimeters. Okay, so how does this autoencoder work? So basically we input a bunch of data here, and then we try to encode it into this latent space. And then using only the information from the latent space, we want to reconstruct or decode the same image. So we do this for B0, and then we do it for the diffusion weighted images separately, like two networks. And then in the testing phase, what we do actually is we take two adjacent slices in the input space here, and then we encode them. And then we take this average in the latent space, and we hope that this average will actually represent, after decoding, represent the average slice in between. And by actually playing with this alpha, we can generate as many slices as we can. And we also do this for the diffusion weighted images. And basically we use this developing human connectome project data, which is a pretty high quality data. And so we use the preterm subject that we suppose that they will be closer to the fetal subjects. So they are between 27 and 37 weeks, but mainly close to the 37 weeks. And the resolution is the in-plane resolution is about one square millimeter, which is close actually to the one that is in fetal, but with a slice thickness here of 1.5 millimeter. So we can basically remove slices and then try to reconstruct them and still be in the range of fetal. So we can either remove one or two. That's actually what we do in the experiments, and then we try to reconstruct them. So we test our network in a four-fold cross-validation, basically remove four subjects, and then we test on the others. And our network is actually composed of four blocks for the encoder and 4 blocks for the decoder, basically convolutional and pooling layers. And and basically so the average is actually performed in this latent space, where we take these features here, these feature maps, and then from one slice, and then from the adjacent slice, and then we average them. Okay, so the results look as follows. So basically here we remove 2 slices and we try to reconstruct them. And this is the ground truth. And basically we reconstruct them by averaging these 2 in the latent space. So this is for the diffusion weighted images for the B0. And then as in the quantitative results, so we plot this box plot here for the mean square error and for the PSNR. So both perceptual loss and also like a direct loss to see how good are we. And we see that our autoencoder here, when we remove only one slice, performs better than the cubic interpolation, also the linear, but I don't show it here. And also for when we remove actually 2 slices, the autoencoder works even better than classical interpolations. Okay, so this is actually for the diffusion weighted images. So for the B1000, where we here I show only removing 1 slice, and yeah, so here are the results by averaging basically from these 2. Okay, so here it's only for one fold so far, where I basically average over all gradient directions. And we also see that it performs better than cubic interpolation. Okay, so then since our goal is actually to test to work on FETAL, so we said maybe why not just taking this network and just applying it to FETAL. And so we basically took the preterm B0 and then we apply it to the our FETAL B0 images, and the results were also okay. I mean, also quantitatively they were okay. So this was for the B0, and here it's for B700, where we actually use the B1000 network to test on the FETAL B700, and it was also generating decent results. Actually, the hypothesis is that we basically think that the network is working pretty well, mainly because it learned from a kind of high quality or also high frequency data. Like we have a lot of details in the preterms. So when it went to a bit easier task in the FETAL, easy in terms of contrast, then it was able to generalize. But of course, we don't want to stop here. So this was only our original signal, but our goal is to go to the diffusion metrics. So we want to evaluate this work on the different diffusion metrics and see how good it performs. And also we want to test it on phantom data. And this is actually what I started to work with. And with this, actually, I needed this variational part of the autoencoder. So basically the second loss that will make the network more generative. And we can see that it's also it's working also fine here. Like when we remove one slice, this is the ground truth and this is the autoencoder. And we also, of course, want to test it on motion settings so that it's more realistic in the FETAL context. So with this, I would like to thank all my collaborators and you for your time and attention.